develop his potential, full potential. He will be a great scientist. So I think that's actually happened. He worked out a lot of difficult problems. There's so many difficult problems I don't have time to explain. I don't want to take up this moment of truth. And so well, he did a really great job, and as a result, he published a number of papers, and he's actually a very well-known student. Whenever I go to a meeting, they will know, hey, how's your magic student doing? Did he graduate? So they all want to hire Jack, and I said, no, not yet. One more year to finish final story. Okay? So there are a number of great personal traits that Jack has that I don't have enough time to explain. But there are two things that I just want to point out. He's actually responsible for organizing Black Party. He's doing a great job, as good as his science. So we really miss him. We're going to be in trouble now. We don't know who to talk to organize this thing. He always found the best restaurant, and the menu, he sent me all the list of restaurant, choice of menu and dessert, and what's good, and it was really great. Another thing is actually, he's very mature, doesn't look that way, but he's actually grown up in Florida, always talking about Gators and football, and ESPN, always sports, but he's very mature actually. But then one time, after 2.5 years, about 2 years after I started my lab, we started lab with 4 people, 2 students, Jack and Ben, and 2 postdocs. And suddenly 2 postdocs decided to leave for various reasons, and I was quite frustrated. Because, as a junior faculty, studied lab, spent so much resource and training and time, and finally they try to get something working on, but then they decided to leave, and I was so frustrated. I didn't tell Jack and Ben about that. I think Jack somehow sensed that I was frustrated. And he came and told me, "Seok-young, I think this is actually a good opportunity. Why don't you focus on my project and Ben's project, then this will be great." So actually, I took his advice wholeheartedly, seriously. I think this is actually, I was like, wow, this is great. Actually, he changed my perspective. So I really work even harder with Zach, day to day, and even night. And so he actually catch up very quickly when we get excited, we solve problem after problem. And now those 2 projects that my student worked on is now the project that fund my lab. Okay? So I really truly miss Jack's leaving, but I'm really excited about his new future. And still think you haven't developed full potential. You have a great future ahead of you.
Thank you, Seok-young, for that touching introduction. I'm going to be taking you guys on a journey today through my graduate work on the structure and function of concentrated nucleoside transporters. Shown here on this first slide is an image of the transporter that we work on embedded in the lipid bilayer, and it's shown transporting pills into the cell since it's a drug transporter in addition to naturally occurring nucleosides. And I hope to explain a little bit better how it accomplishes this feat through the work that we've done here over the past several years. So, something's cut off here. As you all know, nucleosides are extremely important in biology as they serve as precursors for nucleotides and the salvage pathway of nucleotide biosynthesis. And then nucleotides in turn serve as the building blocks for DNA, RNA, and ATP. And there are certain cell types that rely solely upon salvage of nucleosides for their nucleotide biosynthesis. So it's important to be able to scavenge nucleosides from outside the cell. Additionally, the nucleoside adenosine acts as a signaling molecule in such processes as coronary vasodilation, neuromodulation, and the anti-inflammatory response. And then a variety of nucleoside analog drugs exist for the treatments of several different types of cancer and viral diseases. So Since nucleosides are hydrophilic molecules, they require a protein conduit by which to enter the cell. And humans possess two different classes of nucleoside transporters to accomplish this feat. Equilibrated nucleoside transporters allow for the passive diffusion of nucleosides along their concentration gradients in a bidirectional manner that depends upon the concentration gradient of the nucleoside. And then concentrated nucleoside transporters, which is going to be the focus of our talk today, actively transport nucleosides into the cell against their concentration gradients by harnessing the energy of an ion gradient, which in the case of human CNTs is the sodium gradient. So as we already discussed the importance of nucleosides in biology, you can see where nucleoside transporters come into play in providing nucleosides for DNA, RNA, and ATP synthesis, as well as the termination of adenosine signaling, and then most clinically relevant in delivering nucleoside analog drugs to their cellular targets. So humans have three different CNT subtypes with differing tissue distributions and specificities for nucleosides and nucleoside drugs. So ultimately, better understanding the mechanism of nucleoside transport could conceivably lead to the design of drugs that can better reach their cellular targets. Here in this table, I'm just showing some of the major characteristics of the three different CNT isoforms, CNT 1 through 3. CNT 1 is mostly pyrimidine specific, CNT 2 transports mostly purines, and HCNT 3 is broadly specific for both pyrimidines and purines. And then you can see the different cell types where expression levels are particularly high for the three different transporters, as well as the differing sodium to nucleoside coupling ratios. CNT1 and CNT2 are both 1-to-1, while CNT3 utilizes a 2-to-1 sodium to nucleoside coupling ratio. Just as a kind of a review, because most of you should be familiar with nucleoside structure, but here are the major naturally occurring nucleosides, the pyrimidines and purines up in the top row. And then on the bottom row, I'm showing a few representative structures of nucleoside analog drugs, including gemcitabine, which is a cytidine analog that's used in the treatment of pancreatic cancer, zidovudine, or AZT, which was one of the first HIV reverse transcriptase inhibitors that was developed, and ribavirin, which is an antiviral compound used in the treatment of viral hepatitis. So just to give a brief overview of what I'm going to be talking about today. First, I'm going to describe the initial structure determination and functional characterization of a bacterial ortholog of human CNTs. And then I'm going to get into the structural features of both the protein and the nucleoside that give rise to nucleoside and nucleoside drug selectivity. And then finally, I'm going to briefly discuss the role of sodium in the ion-coupled transport of nucleosides. So, since we are a membrane protein crystallography lab, we first sought to solve the crystal structure of a concentrated nucleoside transporter. And since eukaryotic membrane proteins are notoriously difficult to work with, they're hard to obtain crystallizable quantities of protein, and even if you can get enough protein, it's hard to get it to behave well biochemically so that you can crystallize it. We decided to instead go after a bacterial ortholog of human CNTs. So when I joined the lab, as Seok-Young already said, we were just getting started, and we got our hands on whatever genomic DNA we could for different bacterial species. And if they coded for a CNT, we cloned it out. And we initially ended up with 16 different constructs and screened through all of them for expression and biochemical stability as evidenced by size exclusion chromatography profiles, and found that the CNT from Vibrio cholera, or VCCNT, behave the best out of the group. VCCNT is an excellent model system for human CNTs as it possesses high overall sequence identity with human transporters, with particularly high sequence identity within the nucleoside binding site. So prior to our studies, VCCNT had not been functionally characterized. So before devoting all of our effort to trying to crystallize this protein, we wanted to make sure that it was indeed a functional nucleoside transporter. So to measure its function in a detergent micelle environment, which is the environment that we were going to crystallize the transporter in, so we want to make sure that the crystal represents a functional state of the transporter. We measured the binding affinity of detergent solubilized VCCNT for uridine using isothermal titration calorimetry. and calculated the KD for uridine to be 45 micromolar, which is similar to the apparent affinities of human transporters for uridine. We also wanted to make sure that it was capable of transport in a lipid bilayer environment, so we reconstituted our transporter into proteoliposomes and developed a radio-labeled nucleoside uptake assay, where you can add your radio-labeled nucleoside to the outside of the proteoliposomes in either the presence or absence of ion gradients, and then after a specified time course, harvest the proteoliposomes, wash off the labeled nucleoside from the outside, and then count the amount of accumulation of nucleoside within the proteoliposomes. And you see a representative experiment here in the presence of a sodium gradient, so 100 millimolar sodium on the outside of the proteoliposomes and no sodium on the inside. We see significant radio-labeled uridine uptake into the proteoliposomes. But when you have no gradient present, so sodium is replaced with choline chloride, and there's choline chloride inside the proteoliposomes as well, so there's no gradients present, we get negligible nucleoside transport. And then with empty proteoliposomes, so actually they're not they're just liposomes, they don't have protein, we get pretty much no transport of uridine. So this suggests that VCCNT depends upon the sodium gradient for uridine uptake. Prior to our work, the only other bacterial CNT that had been functionally characterized from E. coli was shown to depend upon the proton gradient for nucleoside uptake. So we set up different proton gradients outside across the membranes of the proteoliposomes and found no dependence upon the proton gradient. So our transporter is a sodium-coupled nucleoside transporter, not proton-coupled, just like its human brethren. And a detailed method is going to be published next month in Methods of Enzymology for this experiment. So after biochemically optimizing VCCNT by screening through different detergents, including lipids in the purification, and addition of nucleosides, getting our initial crystal hits and optimizing them, and then trying to solve the phase problem, we initially tried slow met derivatization for SAD phasing, and that didn't work out very well for us, so we shifted over to heavy atom derivatization. We screened through about 40 to 50 different heavy atoms, both by co-crystallization and soaking methods, and we're finally able to identify a platinum derivative that we use to calculate SAD phases to ultimately end up with a structure of 2.4 angstroms of VCCNT bound to uridine, so we crystallize in the presence of uridine. And I want to show just a brief video to orient you to the structure, and so you get an idea of what we're looking at in the remaining figures of the talk. So as you see here, VCCNT crystallizes as a trimer with each individual protomer colored differently here. And we're usually going to be looking at the protein with the extracellular side on top and the intracellular side on bottom. So from within the plane of the membrane. As I stated, we crystallized VCCNT in the presence of uridine. So we saw a uridine molecule bound in the center of each individual protomer, colored yellow here. But typically we're going to be focusing it on just a single protomer, which you can see here in light blue on the outside of the protomer is what we call the scaffold domain. It's involved in forming the trimerization contacts and maintaining the overall architecture of the transporter. And then the colored regions in the center constitute the transport domain, where nucleoside and sodium binding occur. So we're going to be looking at a lot of figures from this view, which shows the nucleoside binding site from the center, like looking out from the center of the trimer axis with the extracellular side on top and the intracellular side on the bottom. So going back to the overall architecture of the transporter, as I already stated, it forms a trimer within the crystal structure. And we're looking at the side of the trimer again, and see that it forms an extracellular knob-like structure on this side. And then on the intracellular side, there's a large basin that penetrates deep into the membrane bilayer, and we expect to be filled with bulk aqueous solvent. So it effectively shortens the distance that sodium and nucleoside must travel to enter the cell. Looking over here, we're just rotating to look up into that basin from the cytoplasm. And then looking at the individual protomer, you can see that's composed of 8 transmembrane helices, 2 of which are partially unwound in the middle of the membrane, as well as 2 reentrant helical hairpins that come in from either side of the membrane, HP1, HP2, and 3 interfacial helices that sit at the membrane-water interface, and as well as this small group of extracellular helices that form that knob-like structure. If we remove that outer scaffold domain that I mentioned earlier and just look at the transport domain over here on the right, you can see that's formed by those 2 helical hairpins as well as the 2 partially unwound transmembrane helices, and that's where we find the uridine in the structure. So looking at the topology diagram, the transport domain is highlighted under these 2 triangular regions, and that's to illustrate the twofold pseudosymmetry possessed by our transporter, the axis of which runs through the center of TM6 here, so that if you were to rotate 180 degrees about that axis, you would have a similar structure. You can pretty much swap these, and you would yield a pretty similar structure, even though the sequence identity for these 2 regions is only about 10%. And this is a common feature that's been observed in several different transporters of this type, since they need to be able to achieve a pseudosymmetric conformational change in order to achieve both the outward and inward facing conformations along the transport cycle. So as I've already stated, we found in our crystal structure that VCC&T formed a trimer in the crystal. So looking at the trimer interface, we see on the extracellular side that it is sealed off by bulky hydrophobic phenylalanines, and on the intracellular side it's also sealed off by hydrophobic residues, suggesting that it can't serve as a permeation pathway for hydrophilic nucleoside and sodium, which makes sense since we saw an individual uridine molecule bound in each protomer. To test whether or not this was a physiologically relevant oligomeric state of the transporter, we generated some cysteine double mutants, shown here and here, the positions, that when you expose to oxidizing conditions, you're going to form a disulfide leakage between these 2 residues that covalently traps the structure in the trimer form. So we did this experiment both in detergent micelles and E. coli, isolated E. coli membranes. And when you expose the mutated, the cysteine double mutant to the strong oxidant copper phenanthroline, we see the formation of this higher molecular weight species in SDS page corresponding to the size of the trimer for both cysteine double mutants, suggesting that it is indeed forming trimer in detergent micelles. And then in E. coli, isolated E. coli membranes, we perform the same experiment, and we see similar results with this band corresponding to the trimer forming. You might notice it's a different scale, and that's because this experiment is done with MBP fusion protein, but it still corresponds to the trimer size. So we think that this is indeed a physiologically relevant oligomeric state for VCC&T, and several of these residues that are involved in trimerization interactions are conserved between VCC&T and human CNTs, so we propose that human CNTs are also trimers. So looking at the nucleoside binding site, we saw in our initial electron density maps prominent non-protein density in between the tips of the 2 helical hairpins and the unwound segments of TMs 4 and 7. And shown here is FOFC simulated annealing map density for the uridine molecule that we eventually built into the density. And as I stated, it's bound in the center of the transport domain, and it's covered on the top by TM6, this long hydrophobic TM6 that it serves as part of the scaffold domain. And if you look down here at the surface representation of the trimer, the mesh representation of the trimer, which is also a surface representation, but you can see that the uridine is bound slightly closer to the intracellular side, but it's kind of pinched off from dissociating into the intracellular side. It's facing that large intracellular basin, but it can't freely diffuse. So we think that our crystal structure represents an inward-facing occluded state, conformational state of the transporter. Zooming in on the nucleoside binding site, we can see all the different interactions between the protein and the nucleoside. several different interactions directly through amino acid side chains, and as well as a few water-mediated interactions. And we're going to look at the energetics of each of these different interactions in more detail a little bit later on in the talk. So since our transporter was found to be a sodium-coupled transporter, there has to be at least one sodium binding site within the structure. So we were able to identify a putative sodium binding site in between HP1 and TM4, and it's octahedrally coordinated by both backbone carbonyls as well as a couple of side chain oxygens and a water molecule with distances that are typical for sodium coordination by protein. You see here that the sodium binding site is near the nucleoside binding site, but the sodium and nucleoside don't directly interact. So we think that sodium serves to kind of bring these structural elements together that are involved in nucleoside binding and complete the formation of the nucleoside binding site, so that nucleoside can in turn come in and bind efficiently. And I'll get a little bit more into that later on in the talk. So this might be a review for many of you, but I just want to go through the basics of the alternating axis mechanism of transport that's expected to be utilized by these types of transporters. So this mechanism assumes that there are 4 major conformations along the transport cycle. And we'll start over here in the upper left in the outward open state of the transporter, where the transporter is assumed to have an extracellular gate here in gray and an intracellular gate down here. And in the outward open state, the extracellular gate is open, exposing the sodium and nucleoside binding sites to the extracellular side. Once they come in and bind, the extracellular gate closes so that they can't freely dissociate back into the extracellular side, forming the outward occluded state. Then a conformational change occurs that positions the binding sites nearer to the cytoplasm in the inward occluded state. And then finally, the intracellular gate opens, allowing for sodium and nucleoside release into the cytoplasm before the transporter cycles back into the outward facing state for another round of transport. So based on some previous structural studies done on the bacterial aspartate transporter GLT-PH, which shares some structural features in common with our transporter, although they possess different overall folds, we proposed a hypothetical transport mechanism that can achieve the conformational change between the inward and outward facing states of the transporter. What they saw in the GLT-PH crystal structures, they were able to capture both the inward and outward facing states of the transporter. And what they saw between the two structures was in the inward facing state and outward facing state, there was a rigid body movement of the binding sites across the membrane, with the outer scaffold domain being held in place while the binding sites moved in a concerted fashion across the membrane. And we think that our transporter could also do something similar, where the transport domain would just have to relocate slightly above this hydrophobic barrier formed by TM6 to expose the sodium and nucleoside binding sites to the extracellular side. And this is relatively shorter distance. This is only about a 10 angstrom different distance that would be required for this change. And they actually saw like a 15 to 20 angstrom difference in their structure. So it's relatively smaller conformational change required to achieve this. So what did we learn from the initial structure? We saw in our functional studies that VCCNT is a sodium-coupled transporter that's active in both detergent micelles and in a lipid environment. We saw from the initial structure that it forms a trimer with 3 independent binding sites for nucleosides, as well as 3 independent transport pathways. We located the nucleoside binding site between the tips of 2 helical hairpins and 2 unwound helices in an inward-facing occluded state of the transporter. We also located a putative sodium binding site that's near the nucleoside binding site, but the sodium doesn't directly interact with the nucleoside, which could possibly serve to stabilize the structure of the nucleoside binding site. And then we proposed a hypothetical transport mechanism that could be achieved by rigid body movement of the binding site across the lipid bilayer while the outer scaffold domain is held in place. So moving on to the next part of the talk, we wanted to see which structural features of the transporter and the nucleoside were most important for nucleoside recognition. So we wanted to probe the individual interactions by essentially mutating the nucleoside instead of the protein. So we used several different nucleosides and nucleoside analogs that have different functional groups to disrupt or strengthen different interactions between nucleoside and protein, measure their binding affinities, and also in parallel we saw crystal structures of several of these nucleosides and nucleoside analogs bound to VCCNT to get a direct visualization of the interactions. So looking again at the nucleoside binding site, you can see that it forms kind of a bowl-shaped structure with phenylalanine 366 serving as the base of the bowl, and the nucleobase interactions localized to one side of the bowl, and the ribose interactions localized to the other side of the bowl. So we wanted to probe each of these interactions directly by essentially removing functional groups from the uridine molecule that we use as our reference. So to determine equilibrium binding affinities for different nucleosides, we developed a fluorescence anisotropy-based competition assay where we use the fluorescent cytidine analog, pyrrolocytidine, which has a pretty strong binding affinity for VCCNT, as our fluorescent reporter. And we can titrate our nucleoside of interest into a solution that contains both the fluorescent nucleoside and VCCNT, and measure the decrease in anisotropy as the fluorescent nucleoside is competed off of VCCNT, and then fit the data to a